Welcome to the second lecture on enzymes. In this particular lecture, we are going to focus on the very basics of enzyme kinetics. Now, what I have done here is to have a very short lecture to introduce you to some of the terms in relation to enzyme kinetics. The details, of course, we will have to revisit when we talk about enzyme inhibition. So you can consider this lecture as a bridge between enzyme activity and enzyme inhibition. The one thing that you must remember here is that you should come to this lecture only after you have listened to the first lecture, which deals with the overview of enzyme activity. So while I am delivering this lecture, I am assuming that you have a basic understanding of enzyme activity. What we are going to do here is to look at the very basics of kinetics. We will define the Michaelis Menten equation. We are not going to derive the Michaelis Menten equation because the derivation is not included in your syllabus and is also not required for the type of assessments or exams that you will face in biochemistry at MBRU, as well as if you are taking any specific licensing exam. So we will define the Michaelis Menten equation. We will focus a bit on a particular term called the Km, or the Michaelis Menten constant. We will derive the Lineweaver-Burke equation, and please focus on this because the derivation and the expression of the Lineweaver-Burke equation is extremely important for you to understand enzyme inhibition. And at the end of this short lecture, we will visit specific terms associated with enzyme kinetics. We will ask you in the assessments some basic calculations associated with enzyme kinetics. So at the end of the lecture, I have worked out a problem for you, and in the self-test, I'm going to upload questions associated with calculations in relation to enzyme kinetics. So, basics of kinetics, defining the Michaelis Menten equation, understanding the term Km, deriving the Lineweaver-Burke equation, and then focusing on specific terms associated with enzyme kinetics. That's the aim of this short lecture. So how do we define enzyme kinetics? So enzyme kinetics is concerned with the quantitative measurement of the rates of enzyme catalyzed reactions and the systematic study of factors that affect these rates. You can understand that we are going to define certain entities in relation to the rates that govern enzyme catalyzed reaction, as well as look at some of the factors that affect these rates. I believe when you look at any biochemistry textbook, at a typical chapter in relation to enzymes, you will see a plot like this. And this is the basic enzyme kinetic plot. What we do here is we add varying amounts of substrate to a fixed amount of enzyme, and the reaction velocity is measured for each substrate concentration and plotted. So we have a fixed amount of enzyme to which we add varying amounts of substrate. So the substrate concentration is varied, and the reaction velocity for each substrate concentration is plotted. Under ideal experimental conditions, the resulting curve takes the form of a hyperbola. What is a hyperbola? A mathematical function in which the values initially increase steeply but eventually approach a maximum level. So as I increase the substrate concentration, the rate increases steeply, and then at some point we reach the maximal velocity. And this fact can be explained by the fact that we are adding a fixed concentration of enzyme and varying the substrate concentration. So at some point in time, all the enzyme active sites in the reaction will be completely saturated with the substrate, and therefore there will be no change, or more precisely, no increase in reaction velocity. So that's what this specific curve tells you. Now let us look at this curve. So what we have done here is we have taken a fixed concentration of enzyme, let us say a hypothetical enzyme E, and to this enzyme we gradually add increasing concentrations of substrate. Of course, initially the reaction velocity will increase, but at some point in time all the enzyme active sites in the reaction will be saturated with the substrate such that we will not observe any increase in the reaction velocity. And from this curve we define the first kinetic parameter, and which is the most important kinetic parameter, which we call the Michaelis Menten constant, or sometimes in the books it is referred to as the Michaelis constant, popularly known as the Km. So what is the Km? The Km is the substrate concentration at which the velocity is half the maximal velocity attainable at a particular concentration of the enzyme. Remember that while you are asked to define the Km, this particular part of the definition is very, very important. So if you do not write attainable at a particular concentration of the enzyme, you may lose marks. Now, this particular plot helps us to derive the Michaelis Menten equation, where the initial velocity is equal to the product of Vmax into the concentration of the substrate divided by the sum of the Km and the substrate concentration. And as I told you, in our course, we are not going to derive this particular equation. But if you are interested, there are textbooks like that by Leninger where this particular equation has been derived, and you can have a look at it and mind it if you are interested. So I'm not going to stress that you derive this equation, but if you are interested, you can look at the derivation of the Michaelis Menten equation. At this point, I would like to highlight that since we will be asking you questions on this equation, questions that relate to calculations, you should remember this equation at all times, especially for the purpose of your assessment. So, what is the importance of Km? If I have a numerically small or a low Km, it reflects a high affinity of the enzyme for the substrate. Why? Because a low concentration of substrate is needed to half saturate the enzyme, that is, to reach half maximal velocity. Similarly, if I have a numerically large or a high Km, it reflects a low affinity of enzyme for its substrate because a high concentration of substrate is needed to half saturate the enzyme. Please, and I am reiterating, please remember these two implications. Many a time we will give you an enzyme and a list of substrates, and I will ask you which specific substrate has the highest affinity for that specific enzyme. What you need to do there is to look at the value of the Km. The substrate which has the lowest value in terms of Km has the highest affinity for the enzyme. Similarly, the substrate which has a high value or the highest value in terms of Km has the lowest affinity. So if I want to have a general expression in my mind, I can say that Km is inversely proportional to the affinity existing between the enzyme and the substrate. A low Km indicates high affinity. A high Km indicates low affinity. And this particular expression will also be used when we talk about metabolic pathways and discuss the concept that we mentioned in the first lecture of enzymes in relation to isoenzymes. For example, the affinity of hexokinase and glucokinase for glucose. Hexokinase exhibits a low Km for its substrate glucose in comparison to glucokinase, which exhibits a high Km for glucose. From this, we can conclude that glucose has a high affinity to hexokinase but a low affinity to glucokinase. So the concept of Km and its implication is very, very important. Now we talked about the Michaelis Menten equation. The problem with this equation is that the direct measurement of the numerical value of Vmax and therefore the calculation of Km, because Km is equal to half Vmax, often requires impractically high concentrations of substrate to achieve saturating conditions. So if I want to calculate the Km experimentally, I will require impractically high concentrations of substrate in order to reach the maximal velocity or in order to achieve complete saturation of the enzyme. So, a linear form of Michaelis Menten equation is required which can circumvent this difficulty. So what we will do here is to look at this equation. We then invert this equation. So basic arithmetic, if I invert it, I get this particular expression. And then I factor it, and I can arrange this particular equation in this particular form. And then I simplify this, and I look at it as an equation for a straight line. And the equation for a straight line is y is equal to ax plus b, where y for us is 1 by the initial velocity, x is equal to 1 by substrate concentration. So if I plot the inverse of reaction velocity as a function of inverse of substrate concentration, I will get a straight line like this, where this particular point will give me the value of the Km. This particular intercept will give me the value of Vmax, and the slope will give me the ratio between Km and Vmax. And such a plot is called a double reciprocal or a Lineweaver-Burk plot. So using the Lineweaver-Burk plot, we can circumvent the difficulty which is posed by the Michaelis Menten equation, which I mentioned earlier. Please focus on this particular plot, because this plot is very, very important when we talk about enzyme inhibition. I will revisit the plot, but please look at this particular derivation very carefully while you are studying enzyme kinetics. As I told you, this is a very short lecture, so we will just revisit some specific terms in relation to enzyme kinetics. The first is the turnover number of an enzyme. So for a pure or homogeneous enzyme, turnover number is the Vmax, or the maximal velocity, divided by the moles of enzyme present in the reaction. The second term is called catalytic constant or Kcat. For a homogeneous or pure enzyme, if the maximal velocity for that specific enzyme is divided by the number of active sites present in the enzyme, we get the value of Kcat or the catalytic constant. Next, we have the catalytic efficiency, which is best expressed in terms of a ratio of two kinetic parameters. And these two kinetic parameters are the catalytic constant and the Michaelis Menten constant or the Km. So the catalytic efficiency is the ratio of Kcat to the Km. I will urge you to make a note of this formulae so that when you are asked problems in an assessment, you can use them effectively. We come to the end of this short lecture. So what we do here, we take a particular problem. So look at it, a particular enzyme which is being studied by a group of students has a Km value, and the value is mentioned here in molar. The students study this enzyme with an initial substrate concentration of 0.055 molar. At 1 minute, 7 micromolar of product is generated. What is the amount of product after 10 minutes? What is the Vmax for this enzyme being studied? For such problems, the first order of business is to look at the units. As I told you, for solution problems, you can convert it to a unit that you are comfortable with. So here I have converted 7 micromolar to molar. I substitute that in the Michaelis Menten equation to get this expression. And from this expression, we will get the Vmax in molar per minute. And then very simply, we can calculate how much product will be formed in 10 minutes. So I will upload some sample problems for you to practice, but this is a very, very important example where I have shown you the importance of knowing the Michaelis Menten equation. Now try this on your own. The substrate S, so it's a hypothetical substrate, can be catalyzed by the enzyme E. The turnover rate of the enzyme is mentioned. Then determine how long it will take E to cleave S. So here I have mentioned the Kcat, which is the turnover number. The hint for this problem is the number of substrate molecule each enzyme site converts to product per unit time is the Kcat. So try it on your own and let me know what the answer is, of course. But if you have any difficulty, we can go through this problem during our in-class session. So that brings me to the end of this very, very short lecture. In our next lecture, we will talk about enzyme inhibition, where we will focus again on the Lineweaver-Burk plot and try to understand the different modes of inhibition. Thank you for listening.